All right, so lecture 8 in the series is focused on the genome and its properties. We now have many data types available to us that measure different functionalities of genomes, and it's become clear that there are many. And I think it's pretty clear that the genome is not just described in terms of a base pair sequence and its three-dimensional annotation. Three-dimensional configuration, I should say, which would be kind of like a four-dimensional space. There are many more dimensions to this, and therefore we talk about the metastructure of genomes because we don't know how many dimensions there really are to genome function. And at the beginning of the chapter in the book, there's a quote to a physicist that said that we live in a four-dimensional bubble, space and time, in an 11-dimensional universe. So the physicists, I guess, think of the universe as being 11-dimensional. We don't know yet how many real dimensions there are to the genome, but there are many. Therefore, this term metastructure of the genome. Okay, so we'll talk a little bit about the concept of a metastructure, and then I want to talk about operons and the arrangement of transcription units. So a transcription unit is a piece of DNA that gets transcribed. We used to think of that as a gene, but now with multiple transcription start sites, you can get many transcription units from the same gene. So if you're a computer scientist, you can think of this as an independently addressable piece of information in the memory that the genome represents. So there are many more transcription units than one would think. You can identify these transcription units and how they're being expressed in a condition-specific manner and how transcription factors orchestrate that. And based on all that information, you can build a workflow that leads to the reconstruction of regulons, and many regulons form a stimulon, and ultimately we would be able to reconstruct the entire transcriptional regulatory network, which has not been achieved yet for any organism, but progress is being made. So TRN stands for the Transcriptional Regulatory Network, and to build that, in E. coli, we have to figure out what the functionalities of the sigma factors are, which are the variable unit of the RNA polymerase, and then the transcription factors themselves. And at the end, I want to mention the challenge that's associated with polyomic data integration, which is substantial. And now this is being recognized by the NIH and other federal agencies, companies, of course, and so forth. And this is now called big data. This is big data analysis. How do we take disparate forms of data and build a coherent whole out of it? And this is not at all confined just to biology. This is being found now in every field. And this is probably one of the most exciting areas to go into as a student. Okay. So there is more to the genome than its three-dimensional configuration and its base pair sequence. So as we talked about, the genome sequences started coming out in the mid-90s. There was annotation to them based on the sequence published, but then a lot of different data types started coming out to characterize that sequence. And here's just a snapshot of that. There is the sequence itself. There is some structural information associated with this. Sequence leads to peptides, leads to structure. There is the operational information, what is being expressed under different conditions, how is the genome operated. And finally, there is the functional annotation we are familiar with that speaks to the function of each of the open reading frames. And as I have mentioned in the reconstruction lectures of metabolism, that annotation is incomplete. There's much more information in the bibliome about the functions of the gene products. So a lot of information, a lot of different points of view, and clearly a very complicated situation. So here is one example that details all of these data types. On the top left We have information like the sequence. We have TSS, the transcription start sites that can now be measured. You can specifically sequence the first 30, 40 base pairs of a transcript and find out exactly where transcription starts, so you can identify the transcription start site. And this has led to the discovery that there are multiple transcription start sites, often upstream from a gene. And then we have in there RNA-seq and peptide mapping. So these are data types you're familiar with. There are other data types you can map onto the genome. And on the right, we see some computational data types. For instance, you can calculate the thermodynamics of unwinding as a function of the base pair sequence. There are the termination sites and so forth. All of this data has to be integrated together, and that's shown in that triangle below. And out of that come numbers like the number of transcription units, the number of, say, TSSs per transcription per promoter, and so forth. And so you build tables like the ones that are shown down there that represent different properties of that genome. So here is a detailed example, and let's look at the DNA icon there on top. You see the TSS, which is the transcription start site. That's where the RNA polymerase starts to make the transcript. Upstream of that are, in this image, two binding sites, one in green and one in kind of orange color. In bacteria, these are the -10 and the -35 box, where the RNA polymerase and the sigma factors touch and bind the DNA. Then you see the RBS, which is more in purple, I guess, pinkish color, the way it projects there. That's the ribosomal binding site, so that's where the transcript binds to the ribosome. And then there is the translation start site down from that. And there's a stub of DNA between the transcription start site and the translation start site that's called the 5' UTR, the 5' untranslated region, which actually turns out to have a lot of function. You can have hairpins form in there that will stabilize the transcript. You can have hairpins form that actually mask the RBS so it doesn't get translated. All sorts of events that can happen in that 5' UTR region. And then, of course, downstream from the gene that we don't see here is the 3' UTR that's after the stop codon, and that's often transcribed, and that can also have a function. So pretty complicated situation when you start looking at it. And what all these panels below here show are properties of a particular genome, in this case, Thermotoca, in the context of that general icon shown on top. For instance, to the top left, we see the actual -10 and the -35 boxes, and these are histograms of how the different promoter sites are occupied in the cell. And to the right of that is that calculation of the thermodynamics of unwinding. And you can see that becomes very favorable exactly where the DNA opens up. So it's easy for the RNA polymerase to open up the double helix and so forth. So a lot of properties that are associated with these characteristics of the genome. And astonishing that this actually functions for thousands of transcription start sites simultaneously, all wrapped up in a volume that's less than a cubic micron. It really makes you wonder what the physical picture here is. Maybe talk about that later on. Okay, so that's the general arrangement of a bacterial genome, what the different properties are. And like these histograms showed in the previous slide, we can actually measure these on an operon by operon basis in a given condition as well. So let me just show you this picture here. This shows the arrangement of the first operon in the E. coli genome, the one that's immediately, I guess, clockwise from the origin of replication. So the top line there shows the ribosomal binding sites that have been found. The strip below that shows the transcription level during exponential phase. Very uniform transcription of this stub of DNA. Right below that is the expression level in stationary phase, and you see that's not even. There's a kink there in the middle of that thing. And then we see there's transcription start sites, the TSS. There are 3 of them upstream from this operon. But then there are 2 transcription start sites in the middle of that operon. Before we thought of this as one unit, but now we know that it is not. And below in green, we see the transcription units themselves. So there are 5 transcription units predicted to come up of this operon. We used to think of always as one, polycistronic situation. Just transcription started and all the genes were transcribed. But now we know that's not the case. There are 5 independently addressable pieces of information in that stub of DNA. And then at the bottom, we see the gene map itself, and the lowest strip in there shows the discovered RNA polymerase binding sites. Now let's look at this a little bit more. So the RNA polymerase can take on different forms depending on which sigma unit is bound to it. And sigma 70 is kind of the housekeeping sigma factor, and sigma S is the stationary phase or the stress sigma factor. And the binding affinity of these 2 alternate versions of the RNA polymerase is different. So you see in this slide, green is the binding state in stationary phase, and the reddish color is in exponential growth phase. So you see in the exponential growth phase, the RNA polymerase really only binds to the first binding site here and leads to this uniform transcription of this stub of DNA. But if you look at the stationary phase, the green bars, the sigma S version of the RNA polymerase leads to initiation of transcription at site 2 and 3 in this operon. And you can therefore see that site 2, because transcription starts there, leads to this non-uniform transcription of the operon. There's that little kink there that comes up, and that's because that is being separately transcribed in the stationary phase. So this is like an operational annotation. It tells you how the genome, how it's being used under different conditions, how these transcription units that are the independently addressable pieces of information are differentially used based on different environmental conditions. So much more complicated than we thought, even on a bacterial genome. Now, it turns out that all this information is now available on a genome scale. We can do, RNA seq to look at the expression level. We can do chromatin immunoprecipitation of the RNA polymerase and find out where it's bound to the genome under different conditions. We can do the same thing with transcription factors, so we can really get information about how these binding proteins are located on the DNA under multiple different growth conditions. And if you do that, you get a lot of data from that. And that has led to a workflow, like the one shown here, that reconstructs a Regulon. So multiple operons that are coordinately regulated by one transcription factors are called the Regulon. And this here shows LRP, the leucine responsive protein in E. coli. So if leucine is in the medium, this transcription factor reacts to that. It senses that and reacts to it and expresses the gene differently. So the first thing I've got to do is to elucidate these components. What is binding where? Where does LRP bind? When does the RNA polymerase bind relative to LRP being bound, and so forth. So you get this component list and the binding states under different conditions. Then based on that, you can start to do reconstruction, and that's what the second box in this workflow shows. And these are the regulatory modes. So the first one are the independent ones, where LRP doesn't do anything. The second one is where LRP enhances the natural regulation of RNA polymerase. And the third box is where it counteracts it, where it puts it in check somehow. And then in the third box, what you do is you look at the function of the genes that are being regulated. And then you start to look at network motifs like the one shown here. So all the genes that may be associated with the import of a nutrient or the metabolism of a nutrient become coordinately regulated. And that's what's indicated there by transport E for enzymes that are utilizing it. And you can have a particular metabolite inside, say like leucine, can regulate all these different processes in a coordinated manner. We'll get to that in a moment. And down at the bottom here is a little box here that just describes the states of this regulon, whether Lucian is there in the environment or not, and so forth. But there is no model yet for this. We don't have mathematical models. And this is what I was indicating earlier. It is not clear how you actually build a computational model for the transcriptional regulatory network. For metabolic networks, we can do flux balance analysis. We all know what that means. Or if you have kinetics, we can simulate time-dependent changes. But how do we do this? So at the very bottom, left, right, I guess, to this slide, we show 3 different approaches to computation here. One is just steady state. Is the operon being transcribed or not, plus, minus. In the middle icon there would be a dynamic response. Something changes and the expression goes up and then reaches a new state. And then the box furthest to the right shows a stochastic effect, because every cell in a culture is doing slightly different things. If you start looking at a single cell level, you start to deal with stochastic effects, because there's only one binding site, either it's bound or not in that particular cell. But you can get a population average if you want. But if you want to look at a single cell, you may have to do stochastic simulations. So steady state models, dynamic models, stochastic models have been built to describe this phenomenon. Each one of which has its advantages and disadvantages. Now, you can do this for a stimulon. So all the families of amino acids are coordinately regulated. LRP, the Lucian responsive protein, is one of the main transcription factors associated with that, but there are others. There's the ARG-R that responds to arginine, and others. And so you can go through this process that I showed you, that 4-step workflow, and you can figure out the network motifs for each one of those regulatory modes. And the bottom line of such a study is shown there to the lower left, where it shows how transport, if amino acids is available, biosynthesis, if it's not available, it has to be synthesized, and utilization is coordinately regulated. So all the gene products in these processes are coordinately regulated. And that coordination is different whether the amino acid is a signaling molecule, like leucine, it's not just a nutrient, it's a signaling molecule, or whether it's a nutrient, so if it's growing on glucose and this is available or not, it basically shuts up biosynthesis and just picks it up and uses it, and that's what that middle line there shows. But some of these amino acids can also become the primary substrate. So the regulon has a different state based on this regulatory logic, depending on the operand that the cell finds itself in. So this regulatory logic can now be reconstructed on a genome scale. Not yet quantitatively modeled. Well, there are approaches to doing it, but I think the jury is out on what is the best way of doing that. And the best way probably depends on what the use of the model, what the intended use of the model is. If you look at global transcription factors, this coordination turns out to be remarkably exquisite. So FIR is the transcription factor that senses iron. And its full regulon has been recently elucidated, and it shows its, of course, has a role in iron metabolism, the import of iron, the formation of the iron-sulfur complexes. It has a role in energy metabolism. I think we'll talk about this later, but a lot of the enzymes in energy metabolism need iron, so that there is some coordination needed there. It actually has an effect on DNA synthesis, and also on a biofilm versus a planktonic lifestyle, that shift whether it's growing freely in solution or attached to a surface. So some of these globally acting transcription factors are just adjusting things a little bit here and there in a coordinated manner. And you can imagine describing this mathematically in the context of a genome scale model will be quite a challenge, because you may have 10, 12, 15 of these globally acting transcription factors. Each one of them is trying to push things around a little bit. Kind of the way special interest groups are affecting legislation in the United States. This tug of war to kind of balance the system perfectly. Although one might argue it's not perfectly balanced when it comes to legislation. But here, for highly evolved cells, this process is honed to achieve a perfect balance relative to what the evolutionary history of that organism was. Okay, so there are some of these genome scale networks that have been built, and the one for sigma factors in E. coli has been published. And this was a heroic effort that took many years. And just briefly, the panel A here shows what the sigma factor does. It binds to the RNA polymerase, and that gives the RNA polymerase binding specificity on a particular promoter based on the -10 and 35 box we were talking about. When transcription starts, when RNA polymerase goes down the DNA, the sigma factor is released. And then, of course, when transcription is terminated, the RNA polymerase is released and the process can start over again. So what's shown in slide panel A here is location data obtained by chip methods, so chromatin immunoprecipitation on a hybridization chip, something we're not going to explain in detail here. But you can do this for the RNA polymerase and the sigma factors individually. So if you overlay these two, you can see which version of the RNA polymerase is binding to each transcription factor for every condition that you test. And then you can map onto that transcription start site data that you also obtain. So you now know where a particular version of the RNA polymerase starts transcription on that operon, and so forth. So you just layer data type after data type on top of one another until you can elucidate the full network. And that's shown with this unwieldy picture there in the middle. So in the middle of this kind of cylinders that go into the diagram are the 7 transcription factors in E. coli, and each one of these blow-up circles show all the genes that are regulated by a particular sigma factor. And you see there are many of these sets that are regulated by more than one sigma factor, so there is redundancy in that system. And if you could take each dot at the end of these conically shaped objects there, you could see the individual operons. And in fact, the one on the top there is the one I started the lecture out with. That's the tryptophan Regulon. And you can see there the S and the D version of the sigma of the RNA polymerase, and how it calls out the transcription units exactly the way I described at the beginning of this segment of the talk. So that information is now actually available on a genome scale. So this is an incredible amount of information that's available. So that's the sigma factor itself. Then we have the transcription factors, and this is a similar diagram, probably generated by Cytoscape, where you can see the transcription factors. Here are 6 global ones shown, and then all the target genes are shown in these purple dots for each transcription factor. And you can see many genes can be regulated by more than one transcription factors. And I think that the red box there indicates that 5 transcription factors can actually affect the expression of that gene. So this will be the transcription factor network that can now be reconstructed. So that's kind of where this field is. We have the data types, we have reconstruction workflows. We can build networks like this on a genome scale. How they're used to then compute phenotypes is unknown. And it is unlikely that that will be done only in a bottom-up fashion. There's probably a top-down approach to this that needs to be implemented at the same time. Now, before I leave this part of the talk, I want to say a little bit about synthetic biology. So as you might imagine, the metastructure concept basically tells you that every promoter upstream of every operon is very complicated, and even the operon itself is very complicated. And we keep discovering things about these elements on the DNA and how they interact. And they're full of surprises. So how can you engineer if you don't know all of these things? Because you will always run into surprises. So therefore, this approach has been developed in the synthetic biology community of refactoring the operons. Now, refactoring, I think, is a term that comes out of computer science when you refactor a code. So this is a way then to break down these members of the metastructure into individual pieces and reassemble them yourself on a piece of DNA, where you know all the functions and how they relate to one another. That synthetic construct may be not as good as the natural construct, natural piece of DNA that has developed all these intricate interactions, but at least the way you laid it out, you think you know its functionalities. So if you want to design cellular function kind of from the DNA up, you try to do it this way. You take all these elements of the metastructure, line them up in a way that you want them to function, and then you synthesize that DNA, make the construct, and put it into the cell, target cell of interest, and you try to boot up that function. So that's how synthetic biology deals with all the unknowns associated with the metastructure of a genome. All right, so the last topic I want to address is the grand challenge that emerges from these considerations. All this data is flowing faster and faster, cheaper and cheaper, better and better resolution. What do we do with it? And therefore we have this grand challenge of big data. In this class we call it polyomic data integration. How do you make sense out of multiple disparate data types? And this is a quote to Sidney Brenner I really like. Just read it out. We are drowning in a sea of information and thirsting for knowledge. Most biology today is low input, high throughput, and no output. And I don't know, this quote was attributed to him maybe 2007, 2008. I don't know if you know Sidney Brenner. He lives part of the year here in La Jolla. He has a position over at the SOC. He comes to campus once in a while and is, of course, a Nobel laureate from 2003, I think. Just such an interesting gentleman to talk to. He's so perceptive and always exciting to talk to him, even if he's now probably 84 years old or so. And he puts out these comments, these very controversial comments. Like one of his favorites, that is probably not written down anywhere, was, I've discovered the gene that causes obesity. It's the gene that opens the mouth. So he will say things like this, which are kind of very interesting. But the second part of that statement is very important. So when we had all these high throughput methods in the mid-2000s, people just did a lot of experiments without much forethought. And that's what he means by low input. People just generated data. Metagenomics started based on the fact we could sequence microbial communities. So let's just sequence them. This tooth versus that tooth in a person. Why? Well, we can do it. Let's see what we can find. So there was not much forethought often put into those experiments. But the throughput was high. You got a lot of data. But the output, in terms of biological understanding and knowledge, was low. Actually, I think he says no output, but it was low. But in the last 7 years or so, the field is getting better, and obviously the kind of models we talk about in this class make sure that you have output, you have interpretation of the high throughput data, like I was going through. And now, as we'll talk about later in the class, we can use models for prospective design. So the input is not arbitrary. We actually design experiments. And then we do the high throughput stuff, and then we interpret again with a model. So a good genome scale model can fix the low input shortcoming and the no output shortcoming. So let me just show you a couple of things that have appeared in the literature in this regard that are interesting. So here are some timelines. Let's look at these bar charts here that are color-coded. So this is pre-next-gen sequencing. It's the first column there. And then now, so 2011 when this was published, and then the projection for 2020. And the color coding corresponds to this workflow of experiment, doing the sequencing, the assembly, and then interpreting the output. So before we had next-gen sequencing, all the work was in the blue box there. It's supposed to get the sequence. You see that blue box is becoming smaller and smaller and smaller. And the orange box is getting bigger and bigger and bigger. The challenge associated with interpreting the data is become bigger and bigger. And in fact, let's see, what was the price of sequencing the first genome? $1 billion or something like that? A couple $1 billion maybe. Now you can get your genome sequenced for the order of $1,000, say. But how do you interpret that information? Well, it's going to take a lot of time and very expensive. So people have published papers with titles like the $1,000 genome, the $100,000 interpretation. So for bioinformatics, for modelers, people that want to work with these big data sets, the future is theirs, and that's that orange box over there. How do we do such integration? This challenge became clear some years ago, and in fact, shortly after Jenny's 2003 review, we can map these data types into a larger scale reconstruction, but you have to interpret the interactions of the data. So Andrew Joyce, who was a student here in bioinformatics in the mid to late 2000s, prepared this review. It's actually a widely cited review, as I realized. And this talks about what happens if you have two omics data types and you integrate them, what you can learn from them together than being apart. But when you have 5 different omics data types, it's not enough to do this in a pairwise fashion. You have to do this, with all of the data types. So it became clear then after that the workflows got heavier and heavier to implement in terms of analysis relative to data generation. And that led Karsten and Zangler here at UCSD and myself to just formally cost out the workflow. What does it cost to implement a workflow with many different data types and many different data sets? So N on this diagram is the number of data sets you have. You may have 10 expression profiles or 100 or 1,000. And N speaks to the different data types. Could be transcriptomic, resequencing, proteomics, chip, whatever. Disparate data types. And what we try to figure out here is what is the relative cost of generating the data versus interpreting the data. And that's the y-axis on this plot. It's the fraction of resources that has to go into basically analysis here. So when this axis goes above a half, you have to spend more than half of your resources on the data analysis and less than half on the data generation. And you can see there, based on this workflow, it's just costed out, that if you have 4 disparate data types, and once you're up to just 3 or 4 data sets, maybe 4, in that situation, the resources that go into the analysis will exceed those going into the data generation. And this is very hard for many people to swallow that have built these big sequencing centers and so forth. Although the realization is now, I think, becoming widespread that this is the situation. And a lot of sequencing and other data types now are being commoditized. So this is a grand challenge, and the genome scale models that we teach in this class are big enablers of making this happen. There are more and more papers coming out that show how multiple omic data sets can be integrated in a coherent way to get a good output from the process. All right, so the summary then of this lecture is that we now have multiple omics data types that can be mapped against a sequence. So the genomic sequence is the template against which you map those data types. By doing that, you can get a lot of functional information about the sequence, not just where the genes are, but when they are expressed, when they are not expressed, are there hairpins on there or not, is there a binding site for a transcription factor upstream from the gene, is the operon fragmented, on and on and on and on. And having all these data types and trying to integrate them has led to the realization that genomes have a high dimensionality. Who knows what it is? But it's much higher than just the base pair sequence and the 3-dimensional arrangement of the genome. So we have now a concept that is still maybe nebulous or ill-defined, maybe not nebulous, but not fully defined, that says that we have a metastructure here that is of some high dimensionality, and there's a lot of information in this simple molecule, or simple, I should say, the single molecule. Obviously very complicated. So if we can measure the state of that metastructure on the multiple different conditions, we can get data types that will lead to the reconstruction of the transcriptional regulatory network. Some people think of that as the operating system for the genome. So we can measure all these data types under multiple different conditions, and we can see how the genome is being utilized under different conditions. And ultimately, the logic for that is embedded in the transcriptional regulatory network. We can now reconstruct it. We can get its topological features. We can get some of its detailed features, but we still don't have a computational model for it that answers all the questions that we would like to answer. Okay, so with that, we'll end this lecture and end this part of the class that is focused on network reconstruction, and we will move on to the mathematical representation of reconstructed networks in the next lecture.